Okay, so welcome to EdPuzzle number 5 on transcription. You're probably thinking, well, what the heck is that, right? So up to now, we've mostly been talking about how to copy DNA, right? The process of DNA replication, which occurs during S phase, right, of the cell cycle in cells that have been given the go-ahead to divide, either through mitosis or meiosis. Transcription is what's happening for most of the rest of the cell cycle, right? We're not copying our DNA, we're not moving it through cell division and mitosis, but rather we're going to actually read it and then make that protein product that's coded for in our genetic material, in our genes, right? And so you're essentially going to read those instructions and then make whatever it says, right? And so that's a two-part process that we call gene expression in total, and it's transcription plus translation. So in class, we've been talking a bit about translation, the process of taking a working copy of the instructions from our genetic material and using that to make protein on the ribosome. So that's a whole separate process that we're going to tackle in class. Here we're just going to talk about transcription, which is the process of making a working copy of our genetic material, right? You don't want to mess with the source code. Make a working copy in RNA instead of DNA. Okay, so where do we begin this process? If we want to read our genetic material and make some protein, well, the first thing we have to do, right, is go where the genetic material is located, which for our cells, right, most of it is in the nucleus, right? Our mitochondria have some DNA, but that's their own DNA actually, and not ours. So our genome is nuclear DNA, and then we have extranuclear DNA. We're going to talk about nuclear DNA. We want to read those directions and make some protein. So we're in the nucleus, that's where we keep our genetic material. And we're going to zoom in on a chromosome. And when we do that, right, remember chromosomes are made of chromatin, and chromatin, right, is histone proteins wrapped around our DNA molecules wrapped around those histone proteins. So if I zoom in even further, I can look at my double-stranded DNA, right, deoxyribonucleic acid, and I've got one gene here, and then of course gene number 2 over here, and gene number 3 over here. And if I zoom in even further, and just this little boxed section, and zoom in on that, I can actually see, right, the order of nucleotides in a sequence there as part of my nucleic acid strand, my DNA strand. And of course, it's double-stranded, right? And our bases are complementary, right? We have one purine paired with one pyrimidine, and it's always the same pairs together so that they'll hydrogen bond correctly across and hold those two strands together. So if I'm zooming in on my DNA here, we also notice, right, that our strands are heading in the opposite direction, right? So one strand is 3' to 5' to 5', to 5', and the other one, the complementary strand, is 5' to 3', right? So you have a 3 to 5 and you have a 5 to 3, right? And I'm going to ask you guys a quick question here about what's the difference between the 5' end and the 3' end. Okay, so zooming in on our section of DNA, in our genes, so this is a sequence of DNA that's going to code for a molecule of some kind. It's usually a protein, and that's pretty much what we'll exclusively talk about in this class is the conversion of genetic instructions to a protein molecule, making a protein molecule. But genes can also code for RNA molecules that we use elsewhere in the body. But for the most part, we're talking about making polypeptides or proteins from our genetic instructions. So you'll notice, though, here it indicates DNA, it says template strand, right? Well, in DNA replication, right, both strands are a template, and then we pair them with the corresponding complementary bases, and we make 2 new strands, and each strand is made up of an old strand and a new strand, right? Well, here we're going to read the instructions, and we're going to read the template strand, and that contains our instructions. The other strand for any given gene is just the complementary strand, and it's not read and converted into protein. So only one strand in any given gene serves as the template of instructions to make protein. And you always have to use the same strand as the template for any given gene. And you can probably see why, right? If you were to read the other strand, you would be reading the gene backwards, right? And you're going to get a different protein, and it's not going to do its job like it's supposed to. So you have to be consistent, always the same strand is the template for any given gene. Now, having said that, for gene 1, maybe it's the top one is the template strand. Maybe gene 2, it's the other strand that's the template. It doesn't always have to be the same strand between genes, but within genes, it's always the same strand. Okay, so the steps in doing this, right? First thing we want to do, again, we're in the nucleus. We've got our gene. The first step in this process of transcription, which is we need to make a copy, essentially a working copy of our gene that we can then use to build a protein. We don't want to touch our source material, right? You don't want to mess with it. You're going to make a working copy instead, and you're going to make that working copy in RNA rather than DNA. So the process of transcription is essentially reading the genetic material in DNA and building a working copy in RNA, a complementary antiparallel copy in RNA to form a molecule that we call mRNA or messenger RNA. And that mRNA is what's going to be used on the ribosome later in translation, which we'll talk about in class. We're going to use that later to make protein. So you can kind of think about you're making a working copy of your instructions. This is very similar to, say, the process of changing formats between text documents, right? The information is still the same. You're just changing exactly what form it's in, right? So we're still speaking the language of nucleic acid, right? We went from DNA to RNA. Those are both types of nucleic acid. So this is kind of similar to converting, say, a Word document to a PDF, right? It's just, it has a different suffix on the end, right? It's got a different, it's a .pdf, right? Instead of a .doc or something like that. It's got a different extension on the end, but you haven't monkeyed with the instructions, and when you open it, it's still in the same language, right? It's still English or Korean or Japanese or whatever it was originally written in. You haven't changed that part. You just changed the formatting, and that's essentially what we're doing when we're making a working copy of our genetic instructions in RNA. Okay, so as I mentioned, you're making a working copy of your DNA, and you're gonna read the DNA with our enzyme complexes, and we're going to write a working copy in RNA. And of course, the reason we have to do this, right, is because our genetic material is in the nucleus, but where are the ribosomes, right? The ribosomes are the protein factories. Right, they're in the cytosol, right? So the ribosomes are in the cytosol or attached to the rough endoplasmic reticulum, both of which are outside the nucleus. So we essentially have to make a working copy, one, to protect our genetic material from damage. You don't want to manipulate that directly. But two, you have to get the instructions from where they're kept to where the protein is actually going to be made, and those are two separate locations. So our RNA molecule really is the intermediate between our genetic material and our proteins, right? So that initial, I'm going to read my gene and I'm going to copy it faithfully, base for base, right, from the DNA into the complementary strand of RNA. That's what we refer to as our primary transcript, aka pre-messenger RNA or pre-mRNA. This is the faithful copy of the whole gene sequence, the whole thing, soup to nuts. In bacteria, they take that pre-mRNA and they translate the entire thing into protein. They don't modify their primary transcript at all. What you see is what you get, right? So what the instructions say, that's what you build. In eukaryotes, right, we actually process our primary transcript. And we have to do that for a couple of different reasons. One, to help get the RNA out of the nucleus, right? Bacteria don't have that problem because they don't have a nucleus, right? So bacteria actually will start translating their mRNA into protein before they're actually done transcribing the thing, depending on how long it is. Eukaryotes cannot do that, right? So we have to process our messenger RNA in order to help get it out of the nucleus and over to the ribosome. We also process our primary transcript to remove certain sections. So we don't end up translating the entire gene sequence into protein because we actually end up modifying and taking some sections out. So our final mRNA molecule, the coding sequence, the actual genetic instructions are usually shorter than the primary transcript. The molecule itself might be actually a little bit longer because we've added some things in order to get it out of the nucleus, and I'll talk about that later on. Right? So again, we're going to make a working copy of our genetic material written in RNA instead of DNA, and then we're going to take that out of the nucleus and stick it on a ribosome in the cytosol. And then we're going to read those instructions, that working copy of the instructions, and the ribosome is going to make whatever, right, whatever polypeptide those instructions code for. And so you can kind of think about translation is a little bit like going from one language to another, because we are, right? We are reading nucleic acid and we're writing polypeptide, protein. So we really are converting it from the language of nucleotides to the language of amino acids. Now the information, of course, is going to be the same, but we are changing how that information is represented by changing what language we're working in. So again, today we're going to concern ourselves just with transcription. We're going to talk about translation and model translation in a candy exercise in class. So today we're just going to be talking about how do we make our working copy of RNA, and then how do we modify it before it leaves the nucleus and goes to the ribosome. Okay, so we have 3 stages of the initial transcription, right? This is making that messenger RNA molecule, right? The faithful one-to-one copy of our gene sequence. And of course they are initiation, getting started, basically. How do we get started? How do the enzymes add on bases to get to the end? That's elongation, right? And then how do the enzymes know where to stop, right? How do they know where the end of the gene is? And that is the process of termination. And we'll talk a little bit about the differences between these processes in eukaryotes and prokaryotes. Eukaryotes, of course, being a little more complicated because, let's face it, that's how we roll as eukaryotes. So how do we start this process, right, of making our working copy? Just like DNA replication, right, we said, how do we start? Well, we need the players, right? We need helicase and topoisomerase and those binding proteins to hold the strands open, and you need primase to make the primer, right, that tells DNA polymerase where to start, right? Specifically giving DNA polymerase a free 3' end of the sugar, right, on the nucleotide. It's giving that end, right, that's available, and DNA polymerase is then going to grab, right, the corresponding nucleotide. It's going to grab the phosphate end and tie those two things together, right, in a condensation reaction to make a covalent bond that we refer to as a phosphodiester bond, right, holding those two nucleotides together. And then it's going to just keep doing that over and over until it can't do that anymore, right? Very similar process here in transcription. In fact, the bond is the same. The nucleotides, though, in this case are RNA nucleotides and not DNA nucleotides, but otherwise the process is almost exactly the same. But we need a few different things, right? Most of the other enzymes are different. So what do we need to start? We need to know where to begin this process. We need to know where the beginning of the gene is, right? In this case, we're going to use RNA polymerase, an enzyme called RNA polymerase 2, because we're eukaryotes. RNA polymerase 1 is bacteria. So we're going to use RNA polymerase 2, which does the same exact job as DNA polymerase, only with RNA bases instead of DNA bases. So it's catalyzing the same exact reaction. The other difference with RNA polymerase, it does not need a primer to start, which is very nice, not to have to hassle with that. It does, however, need to know where the beginning of the gene is, and that is actually a sequence on the DNA molecule itself that we call the promoter. So the promoter is essentially the starting line for RNA polymerase. It's telling it exactly where to start that transcription process. In addition to the promoter, we also need a few other players. We call these transcription factors. And the transcription factors are usually proteins. Essentially, they're coenzymes for the most part. And they bind to the DNA at the promoter to start with. And they are essentially acting kind of like something sticky like Velcro, right, for RNA polymerase, or almost like you want to think of it also maybe like a magnet, right? So RNA polymerase can see the promoter, but it can't actually start the process of transcription without these transcription factors. The transcription factors help open up the double helix so that RNA polymerase can access the gene sequence, so it can read it and add on to 3' end. And they facilitate this whole process. So where the transcription factors are bound to the promoter, it attracts RNA polymerase 2, again, kind of like Velcro or a magnet, and sort of getting it to start there. And then once you have your transcription initiation complex, which is all your players, right? So you have your transcription factors plus your RNA polymerase II. Those things are going to bind to the promoter, which is the DNA sequence. So you can sort of think about it like you have your transcription factors are sitting on top of the racetrack, and the promoter is the starting line. And they're all there. Now you can start transcription. So you have to have the proteins help RNA polymerase do its job, and the transcription factors also bind to the promoter and to the rest of the DNA as transcription happens. And of course you have to have RNA polymerase, whose job it is to add the complementary RNA bases on as it is reading the DNA instructions. And then of course you have to have the promoter, which shows it exactly where to start. So I think it's always useful to have a visual to try to wrap your head around this. So what does this look like, right? So here we have the initiation, right? This is the start. And of course we've got our promoter region right here. Includes something called the TATA box. This is just a sequence that tends to be found in eukaryote promoters. That's really all that is. For our purposes, we don't really need to care too much about that, other than knowing the promoter is that sequence right before the start, right? So here's the starting line of here's where the gene starts, right? Where RNA polymerase is going to attach with the help of our friends, those transcription factors, right? So they're going to bind to the promoter, specifically the region around the TATA box, but not just exclusively there. And they're going to bind there before RNA polymerase comes along. Because remember, right, those transcription factors act like a magnet for RNA polymerase. It's a sort of a come hither, come over here. This is where the fun is happening. This is where we're going to transcribe a gene. And don't you want to be part of the party? So they're basically beckoning RNA polymerase to come over and bind to them, and in the process be able to read the gene, right? Now this is the template strand, is the bottom one, right? In this particular case, so the template, right, is always going to go 3' to 5' in the case of transcription. Why, you ask? Well, I'm so glad you asked that, right? Why does the template have to be in the 3' to 5' direction? So hopefully everybody answered because, right, RNA polymerase has the same limitation as DNA polymerase, which is it can only add on to the 3' end, right? Because remember, it's catalyzing that phosphodiester bond. So it has to have a free 3' carbon on the sugar so it can grab the phosphate, join the two, and make that covalent bond there, right? So if the template is 3' to 5', when I add my complementary bases, right? So I've got a 3' end there, the complementary end is going to be the 5' end. And here we have, right, in the bottom, RNA polymerase has started. Here's the starting point of the gene. I've got my promoter, right, which I'm attached to the promoter, right, and the transcription factors, and I'm kind of riding on top of them sort of like a train track. I'm bound to my transcription factors. I've got RNA polymerase here. We're going to open up our double helix, and there's the starting point, right, which is probably going to be a start codon for folks who are paying attention when we were talking about that in class just the other day. You're going to have your start codon, and again, that's the 3' ends. So the first thing they're going to lay, the RNA polymerase is going to lay down is the 5' end of your nucleic acid. Well, that means that the other end of the nucleotide is the 3' end over here, and now we're off to the races, right? Now we're off to the elongation part of this process because we've right? We've got the starting point, we have all of our players, we've popped open our double helix, and we've added that very first nucleotide. Okay, so elongation, right? Our very first nucleotide is over here on the far left, right, where my little cursor is wiggling around, right? So that's the first one, and then of course then you have a 3' end that's available, right? And we just have RNA polymerases continuing to catalyze that reaction, that phosphodiester bond, right, that's joining all of our nucleotides together. So I have a 3' end, say that 10 times fast. Free 3' end is here, and I'm RNA polymerase, and I'm catalyzing that bond, and so I'm just grabbing those corresponding complementary RNA nucleotides that match the DNA template, and I'm adding them on, and I'm catalyzing that reaction to join those nucleotides together, right? And I'm reading the template strand. So I'm always adding on to that 3' end, right? And this happens pretty quickly, actually. RNA polymerase is no slouch. It moves pretty fast, and transcription happens very, very quickly. RNA polymerase can only, as part of this transcription complex, can only transcribe one gene at a time. However, right, our cells make lots and lots of copies of these enzymes, so you can have multiple transcription complexes reading the same gene, right? They can't be in the same physical location because they get in each other's way, right? But you can have one that's almost done, right? And one that's halfway done, and one that's just started, right? Depending on how many times you want to transcribe this gene, which depends on how much protein you want to make. And that's something we're going to talk about later on when we talk about gene regulation, which is a big factor in that is, hey, how much protein do you want? If you want a lot, you might transcribe the same gene multiple times, right? So RNA polymerase is reading, right, the template strand, because the template goes from 3' to 5', and of course then I'm going to write in the 5' to 3' direction. That means the other strand that goes 5 to 3, right, is the non-template strand in this particular gene, right? If the other strand was the template, right, the promoter would have to be over here, right, because that's the, oops, if my 5' end is here, right, that's the 3' end. So that I would have to start, my promoter would have to start over here, and I would have to go this way, right? So you have to go in the opposite direction, which as we can see, right, I would be reading G, G, T, T, A, C, C, and so on, this direction. That's what I would be reading, and I would be writing the complementary bases in RNA, right? So C, C, A, U, U, G, G, right? A, U, G, and so on. That's a really different sequence than what you're reading here, because you would be reading the complementary sequence and you're reading it backwards. So you're not going to get the same gene if you try to transcribe the non-template strand. Not going to work, right? Now, however, again, another gene, this might be the template strand, in which case the promoter is going to be over here and the whole complex is going to be flipped around and heading the opposite direction. Okay, so how do we stop this process, right? Obviously, you're not going to read the entire chromosome, right? You're going to read to the end of the gene and then theoretically stop, right? Because that's the end of the instructions. I say in theory because, of course, eukaryotes are weird, right? We are just complicated, and that's just how we roll. So in eukaryotes, transcription doesn't stop when RNA polymerase reaches what's known as the stop codon or the termination sequence. Of course not. So essentially, it reads the instruction, the sequence that says, hey, this is the end of the gene, right? That's essentially what this is saying, right? So A, right? This equals end. End of the gene. That's the end. But RNA polymerase in eukaryotes, RNA polymerase 2, doesn't stop transcription at this point. It keeps going. And it actually takes some proteins called termination proteins who recognize this particular RNA sequence in the primary transcript, right, our pre-mRNA, and they're on the lookout for that, and they see that and go, oh, okay, we've reached the end. So about 10 to 35 nucleotides downstream or after, try to write after with this, it's not going to go well. Oh, that's really ugly. Okay, it looks like offer anyway, or otter anyway. It's supposed to say after, so downstream is after. So the termination proteins basically see the sequence and then let it go by a little bit, so to have a little buffer, and then they essentially excise or cut, splice. They cut the pre-mRNA molecule free from our transcription complex. And now I have my pre-mRNA molecule, and then I have my transcription complex, which now that we're done transcribing, it essentially falls apart and can come back together on the promoter to start the process over again. So RNA polymerase 2 in eukaryotes behaves a lot like a runaway train in some ways, or one of those, runaway, big rigs on the freeway or whatever whose brakes have failed. It just keeps going. It blows right past that termination sequence, and it actually takes additional proteins or enzymes, right, to recognize that sequence and then splice the pre-mRNA, cut that molecule free from the sequence in order to stop transcription. In prokaryotes, those little guys who are so much more efficient in some ways than we are, their RNA polymerase actually recognizes the termination sequence in the bacterial genome. It says, oh, hey, look, here's the end. I'm going to unbind, right, from my proteins and from the DNA, and I'm going to just stop doing what I was doing because I've reached the end of the gene. Would that eukaryotes were so efficient and clean like that? Because we're not. We're messy. Anyway, so, but in a way that's kind of okay, because, so essentially we have extra code after the end of the gene on our primary transcript in eukaryotes. And that's actually okay because we're going to add some more stuff to our pre-mRNA molecule before it leaves the nucleus, and we do that in part to help it actually get out of the nucleus. It actually makes it a little bit easier to get through the transport proteins and out those pores in the nuclear membrane and out into the cytosol, which is where our ribosomes are hanging out and waiting to join them to make the translation party, right? It makes it easier to get it out of the nucleus, but there are some other reasons why we add stuff on to our pre-mRNA molecule, as I will get to here in just a second. Okay, so what happens in post-processing, basically what we call mRNA processing. We are still in the nucleus, by the way, right? We haven't left the nucleus yet. So we have our, we finished transcription. We've busted our transcription initiation complex is done, and we have our working copy of the entire gene sequence. So we've copied the whole thing, in fact, a little bit extra, right? As we see here, right? So this is that polyadenylation signal. You don't have to worry what the heck that means. We'll just don't worry about that, right? But here was the stop codon. This is the termination sequence plus those extra 10 to 35 nucleotides, right? That's this bit here. So this is that extra at the end. And then we had the beginning of the gene in this particular sequence. And then here's the start codon. That's the first, this is where mRNA, our ribosome is going to start translating. And then we have the stop codon, that's the end. So this is what we refer to as the coding segment, right? This is where the actual information is. This is what we really, really care about for translation, and we want to protect that, right? So we want to make sure our mRNA molecule gets out of the nucleus, and that's one of the reasons why we add this cap at the beginning called the 5' cap. We also add a poly A tail at the end. This is basically just a whole bunch of adenines. Basically, it's just a bunch of A's at the end. And both of those things help get mRNA out of the nucleus, our mRNA transcript, but they also protect it from hydrolytic enzymes. And remember hydrolysis, right? This is the process of, if you're hydrolyzing a bond, right, you're breaking the bond using water. But these are hydrolytic enzymes, are enzymes that break bonds using water. And these include things like nucleases, whose job it is to break down nucleic acid into nucleotides. Believe it or not, we do have nucleases floating around in the cytosol. One reason for that is to help protect against viral infection. So they will spot basically pieces of RNA or DNA that are floating around in our cells and just cut the bonds, right? Those phosphodiester bonds, and just cut them up. Slice and dice like a bunch of scissors. We want them to do that to viral DNA or viral RNA. We don't want them to do that to our messenger RNA. So putting the caps on the ends helps protect our messenger RNA from hydrolytic enzymes, particularly the poly A tail, because those enzymes essentially start at the end and kind of chew their way up the tail. So how long the poly A tail is actually affects how many times we can translate these instructions on the ribosome. Because as we're going to see when we get to translation, we can reuse, I often do, reuse our mRNA molecule. We essentially translate it over and over and over again. Every time you translate it, you make another protein using the same instructions. If you want to make a lot of protein, that's a really efficient way. You don't have to necessarily make, well, you usually probably make more copies of the transcript anyway. But each individual transcript can be translated multiple times until that poly A tail is eroded, and then you can't use it anymore, and that will stop the translation process. So the poly A tail is, again, there to protect from those hydrolytic enzymes. It helps get the mRNA out of the nucleus. And the 5' cap that's added to the start end, the beginning part of our pre-mRNA molecule, also helps get the mRNA out of the nucleus, and also helps the ribosome attach to the starting end, right, which is the 5' end, right, because that is where we had the start right? That's where the promoter was on our template, right? Was the 3' end, and we added the complementary 5' end when we were reading it. So that therefore the 5' end is the beginning of our pre-mRNA molecule. So some of the main things we do is add a 5' cap and a poly A tail. We also have some extra non-coding sequences, as we can see kind of here in the middle, and this other junk sequence that was the termination sequence plus that extra code. Okay, so the last thing that happens to what was our pre-mRNA transcript, right? Preliminary or primary transcript. Once we modify it, right, it'll be our final mRNA molecule that's going to go to the ribosome and get translated into protein. So we've, to date, right, so far, right, we've added this 5' cap and the poly A tail, and now both of those things are going to help get our molecule out of the nucleus and protect it from those pesky hydrolytic enzymes, right? And help it attach to the ribosome later. The last thing that happens, as we are actually going to splice and dice our pre-mRNA molecule to make our final mRNA molecule. So as you guys noted probably in the previous slide, you notice that UTR, right? This stands for untranslated region. You don't really have to know that, so don't worry about that too much, but it just means we have some extra code. So we have extra code here, and we have some extra code here on the ends, right? Which this we know is that termination sequence plus those extra nucleotides, and then we have some extra code here at the beginning, right? So our start sequence on our gene when we were transcribing was here. The actual start codon isn't until here though, right? So this is our coding bit here in the middle. But you'll notice our pre-mRNA, right, looks a little different. Okay, here's my UTR, and I've got another one over here on the end. Here's my start codon, right, which is going to be AUG, we know. If I can write it. All right, that's the start codon. And then we have these other sections. We have exons and introns and exon and intron. And what the heck is an exon and what the heck is an intron? In yet another example of people not getting the opportunity to name something ever again because they name it in a way that makes absolutely no sense. This drives me insane right along with you guys. Okay, so introns, in an ironic twist of fate, introns are non-coding sequences, right? What I generally call non-coding regions, right? So this is junk. This is junk code. Same with this guy over here. So the introns are not coding sequences, meaning they're not part of the final sequence to make the protein that we want to make, right? They're not part of the final instructions. They're just some junk. We've got to get rid of the junk before we can send this beautiful piece of RNA off to the ribosome. So weirdly, I know it sounds like, okay, intron even has the word in it, right? X like exit, you would think the introns would stay in and the exons would leave. But no, it's the opposite, because obviously we're in opposite land. Anyway, I apologize, but this makes me insane. So the introns get spliced out, and they get spliced out by these really cool enzymes called spliceosomes. And a spliceosome is a protein and RNA molecule complex. And it's actually the RNA molecule that catalyzes the reaction. So a spliceosome is a kind of enzyme. It's a weird enzyme where the RNA is actually catalyzing the reaction and not the protein. We have another example of that when we get to the ribosomes, which we can also refer to as ribozymes because they're an enzyme, but it's the RNA molecule that actually catalyzes the reaction and not the protein. So weirdly enough, but we do know the RNA was probably the first molecule of inheritance anyway, so the fact that it does double duty is probably not quite so surprising anyway. So our spliceosomes are binding based on their structure to certain sequences on the introns. And then, as the name implies, they're splicing. So they actually do cut on either side of the intron. In fact, if I can find where'd my cursor go? Right, so if I'm a spliceosome, I'm going to cut here, and I'm going to cut here, and I'm going to cut here, and I'm going to cut here. It's kind of like the restriction enzymes you guys are working with in lab. I'm going to cut those sections out, and then I'm going to join the exons together by, again, catalyzing that phosphodiester bond between, right, this 3' end on this exon and the 5' end on this exon, such that eventually, right, I get one whole piece of coding sequence, right? And this is the sequence, this is the instructions that are going to be read by the ribosome and made into protein. So that's our final mRNA molecule. I know, it makes absolutely no sense. So we actually take the introns out and leave the exons in. I know, your head probably wants to explode at this moment. I apologize. I didn't name the thing. Okay, so just to quickly recap, right? Last thing. So we've taken our pre-mRNA molecule, right, which is the product of that transcription process, right? And we started with the promoter, and that was the start point. And then we transcribed faithfully added on the complementary base pair using our DNA template, adding it on in RNA till we get to the termination sequence, right? And because we're eukaryotes, RNA polymerase blows past the termination sequence, and it actually needs the help of some other proteins to identify the termination sequence and then cut the pre-mRNA molecule free from our transcription complex, which then falls apart and can be reassembled to start the process over again and make another pre-mRNA molecule. Our pre-mRNA molecule then gets a poly A tail added onto one end, that's the back end. It gets the 5' cap added onto, as the name implies, the 5' end, right? And all of those things, right, help get the RNA out of the nucleus, help it attach to the ribosome, and help protect it from those hydrolytic enzymes that are floating around in the cytosol, wanting to essentially break it down and destroy it. And of course, because of course the cool thing about mRNA is you can reuse it multiple times, but once it gets too degraded, then you've got to send it to the recycling center, the lysosome, and break it down into its pieces and reuse those nucleotides over again. All right, so our pre-mRNA also gets cut or spliced by the spliceosomes so that the introns are removed and the exons are joined together. And then that's our final mRNA molecule. So it's got some non-coding regions, some extra code on both ends, plus a 5' cap, plus a poly A tail. And then that's going to go to the ribosome. So the process of, part of this is splicing, right, we refer to as alternative RNA splicing, and it's alternative because the cool thing about this, so the non-make your head explode part, is that, right, remembering, right, every cell has got the identical copy of, hopefully, of every gene, right? Obviously, not every gene is expressed in every single one of our cells. And we're going to talk about this more exactly how cells decide which genes get turned on and which ones get turned off later when we talk about gene regulation and development. But because you have the same gene sequence, what's cool is you can actually make different proteins from the same gene sequence by treating those coding sections and non-coding sections differently in different cells, right? So maybe in one cell, you have a section that gets treated as an intron, so it gets cut out and removed. And in another cell, those spliceosomes see that intron and say, oh, actually, I'm X type of cell. I am actually going to see that section, and I'm going to treat it as an exon. I'm treating that as a coding section, and maybe I treat an exon as an intron, and I cut that out instead. So essentially the same pre-mRNA molecule can be made into multiple different final RNA molecules, mRNA molecules, by treating different sections as introns or exons in this alternative RNA splicing process. I apologize, I probably just made your head explode. Sorry about that. But it's super, super cool, right? So depending on what type of cell you are, right? A blood cell, muscle cell, a neuron, whatever, a liver cell. Depending on what type of cell you are, that's going to help determine what kind of proteins you need to make. And you can make different proteins from the same set of instructions by treating certain sections as introns or exons and treating them differently in different types of cells. Super, super awesome. And again, we'll talk about this a little bit more later on and get into sort of what that means for us. But it's a really, really great way of being able to be flexible and making different types of proteins, but not actually changing the source code at all, right? The DNA stays exactly the same. It's just the working copy gets spliced and diced differently depending on which cell you're in. And the spliceosomes are going to be different in different types of cells. Now that may be possibly the result of differential gene expression, right? So you may have different versions that get turned on or off depending on which cell you're in. But it's a pretty powerful process. All right, so that is the end of transcription, and we will do more fun stuff with translation in class, putting the two things together. So we've made our working copy of our RNA molecule, and now we're going to send it to the ribosome.